Right, here we go. It is week 4. It is the fall of 2020. It is Monday, and the birds are chirping. The air is clear, no smoke, at least for now. So where have we been? Remember, we completed fertilization last week, and we created at first a zygote when the genetic material mixed together. And then our one-celled zygote split into a two-celled zygote to create an embryo. And I said it was okay. If you forget, you can always call it the Z word, the zygote. But it is technically an embryo at the two-cell stage. That splitting, by the way, is called cleavage. And then we saw our little embryo split again. The cells within it split again and again. It's still the oocyte, though. I mean, this is all happening inside the oocyte. We still have a zona pellucida around there. We have a corona radiata that is degenerated in a way, though. That usually takes off, degenerates away by about day 2. But we made it up to day 4, and we had a little oocyte with 16 cells within it, so a strange little oocyte, and we shouldn't call it the oocyte, of course, anymore, but that's really what it is. But we call it the embryo. And at the 16 cell stage, we renamed the embryo the morula, but it still answers to embryo, it still answers to zygote. All right, and all this was happening while we were moving down the fallopian tubes, because of course the name of the game is to implant our little creature right in the wall of the endometrium, and then it needs to get sucked in to the meat of the endometrium, and we'll look at that in a minute. There's some, how does it move down the fallopian tube, you may be wondering? Well, there's cilia in there that sweep it along, so it's like little brooms in here. I'm sure Dr. Doas went over cilia. And there's also a little peristalsis going on. So we can have some smooth muscle contraction, and that helps push the oocyte toward this little sphincter right here that needs to open. And then it goes kerplunk in this, call it the martini glass, because it's filled with uterine fluid. It's completely filled with fluid in here, so it goes kerplunk. And that's where we're headed right now. Now we've got to do a little molecular biology, so I'm sure you're doubly excited about that. Embryology on top of molecular biology. It's not going to be crazy, though. But we have some segregation going on, because right now we just have a mass of cells, and this mass of cells, let's say the 4-cell stage, they're just a mass of cells. They all look alike. There's no difference between them, but now we're going to start differentiating some of these cells. And the first differentiation or segregation, segregation means that they move, not only do they differentiate, but they actually move to different areas of the cell. That is, they're going to segregate into the intercell mass and the trophoblast. All right, so late in day 4, blastomeres and the morula, they segregate into 2 groups. A group that's kind of in the middle. Here's our little zygote, so we're going to get a bunch of cells clustering here in the middle, and then we're going to get cells clustering around the outside. The ones in the middle are called the intercell mass. Universally, everybody calls it that. That darn Langman, which is the chiropractic board book, calls it the outer cell mass, and he's the only author that does that. But I worry that might show up on boards, so you have to know that AKA, but nobody in the world calls it that except him. Everybody else calls it the trophoblast. Trophoblast is this outer group of cells right here. So far, so good. But how does this segregation occur? So we need to talk about some genes. So genes, of course, you know the deal with genes, right? There's a cell, there's the nucleus. We have chromosomes that live inside the nucleus, and chromosomes are made up of 2 strands of nucleic acid, and together those 2 strands of nucleic acid make up, we call it deoxyribonucleic acid, or DNA. And the genes can have, sometimes they have double appearances like this, so these would be the prophase look. But really, they would be so small we couldn't see them, but they would be all scraggly and all over the place Because remember these chromosomes spend most of their time in inner phase, and that's just a wild and crazy phase. But then they have to double their chromosomes. What are these called, these little arms here? Anybody remember that? Chromatid. So this, and remember we always count the centromere as 1. So this is 1 chromosome. Let's make it bigger. So this is 1 chromosome. That's the centromere. And this is an interphase. But as we have to pass through the cell cycle, if we want to split that cell into 2, in order to do that, we need to do replication here. We need to do DNA replication. That occurs in interphase. And now, oh look, we have double the amount of chromosomes. We have 2 chromatids at this point. But confusingly, this is still 1 chromosome. You always count chromosomes via the centromere here. So that's just basic stuff. I won't go any deeper than that into it. You'll get that in other classes a little bit, but it's kind of assumed, I think, that you know that, because you probably get that in junior high these days. And if you're shaky, there's zillions of YouTube videos on this, the central dogma. All right, now, so why did I start talking about that? Because I want to show you where genes are. So genes live on these chromatids, right? So genes, there may be hundreds of genes, but they're little regions of DNA. Let's say this is a gene right here. Let's say that gene makes your, how about a sodium-potassium ATPase pump right here. And there's another gene right here. Let's say that one makes, how about a aquaporins 2 channel. And there's another gene right here. And that one makes, I don't know, an aquaporins 4 channel. How about a GLUT4 channel? So basically they almost always make protein, right? So genes live on these chromatid, really. But they're the same. There's another gene right here, because this strand, our original strand, was duplicated. So these are all pairs of genes, right? So they're all duplicated. Everybody, I think, understands that stuff. So we got some new genes that in the morula, some genes start waking up. And these genes are going to express stuff via the central dogma. And that stuff is going to tell some of the cells to move to the outside and some of the cells to move to the inside. In fact, the morula, that's the earliest example or manifestation of embryonic gene expression. Right? So let's get a little deeper into that. And there's the central dogma. I mean, you guys know that, right? I don't need to go through that. Where some authors actually include replication as part of the central dogma, but traditionally the central dogma is here's a chromatid, here's 1 arm, right? Because the DNA is double-stranded. So there's 1 chromatid. So that little thing is this little bump right here. Right? So let's blow that up and look at it. This is actually a gene. So this gene, this is broken in half, and something called RNA polymerase reads these little colored things. Those are the base pairs. Remember, a nucleic acid is just really a chain of nucleic acid is a chain of nucleosides and nucleotides. Remember all that stuff? So that is transcribed by RNA polymerase, strangely, into a strand of RNA molecules. So deoxyribose is replaced by a ribose is only the big deal, and we have uracil instead of, thiamine, if my memory serves. But it's a copy of this piece right here, just this little piece, because this is split in half. And those are the instructions those contain the instructions for building whatever our protein. We'll talk about a GLUT4 channel or whatever channel. Let's do the sodium-potassium ATPase pump was the first one I used. So this is the blueprint how to make that. So this needs to be converted into protein. How do you do that? So a ribosome, this hooks up with a ribosome in a process called translation. And this amazing ribosome, every 3 of these code for 1 amino acid. And so it adds amino acids onto this transcript, and you get a twisty protein that is folded just perfectly. In this case, this is our sodium potassium ATPase pump, and it's inserted in the cell membrane and it goes to work. That's the central dogma. So if you're shaky on that, go watch the Amoeba Sisters. Oh, there's other videos on it. There's tons of videos on this stuff as well. All right, that's all I'll say about that. So the human has, of course, 23 pairs of chromosomes, and there's hundreds to thousands of genes depending on what chromosome. Here's another thing much better than I drew it, where these are all, here's a gene here, here's a gene here, this is a chromosome. Let's say it's chromosome number 22. These are all different. These code for all different sorts of stuff. Some of these genes may be asleep. Maybe this one's off. Maybe this one's off normally. Maybe this one's off. Sometimes it's not a good thing to have genes that are supposed to be off turned on, because then they start making proteins that the body might not recognize, and the body's immune system may go, oh, we don't know who you are, we better attack. And that's one of the kind of the theories of autoimmune disease, is when genes wake up that are supposed to be sleeping. All right, so everything I just said there. Sometimes we can get problems in interphase where, look at this chromosome. Here's the original copy of the DNA molecule. Oh, they forgot to put a gene in. For whatever reason, the replication process messed up, which is done, by the way, by DNA polymerase, not to be confused with RNA polymerase, which is good for transcription. But replication is DNA polymerase. So it messed up here. That's called a deletion. And when you, remember we said every 3 of these base pairs are read, and then one amino acid is put. If you're missing one of these, you get what's called a frame shift mutation, and those are really, really bad. That cancers can occur and all sorts of trouble. And there's all sorts of mutations that somebody's supposed to teach you. I'm not sure who, or I'm not sure if you're just supposed to know that stuff, but I guarantee you that stuff's on board, so make sure you're familiar with that. But that's a frame shift. Not too terrible. I mean, I'm kind of stimulating your memories. So this is a human karyotype. These are the 23 genes. Remember, there are, if we talk in pairs, there's 22 autosomes. This isn't every cell. You scratch your cheek, take a cheek cell, take a nose cell, an eye cell, a liver cell, a heart cell. You name the cell, and it has 23 pairs of genes, except for, how about a secondary spermatocyte? Does that have 23? No. There's a couple cells that don't actually have that, but most cells in the body do. But remember, there's 23 autosomes, and then there's 1 pair of sex chromosomes in every cell. So your cheek cell has sex chromosomes in it. All right, and you can look at those sex chromosomes and tell, for example, what is this? Is this a boy or a girl? We can tell XX, that must be a girl, right? If it was a XY, it would be a male. All right, basic stuff. You guys know that. All right, so let's meet this, our first segregation. So we created an inner cell mass, 1 AKA. That darn Langman and his AKAs, he keeps trying to change the world, and it's confusing to everybody. If this gets on YouTube and people are watching, medical students are watching, they don't use Langman, so they don't have to worry about that. But you chiropractors have to worry about that. Inner cell mass, aka embryoblast. Right? This is you. You are an embryoblast. Or you, I'm not going to even say that. You're an inner cell mass. That's what makes, that's going to become you. All right? It's gives rise to ultimately the intraembryonic tissue, or the embryo proper, the 3-layer disc, the trilaminar disc. This is all going to be inner cell mass. And then we have kind of a support system around the little growing human, and that's trophoblast. That's going to change to cytotrophoblast. That's always very confusing. If you ever try to read this stuff, there's a weird AKA called cytotrophoblast, and we'll look at that. But there's Langman and his weird AKA outer cell mass. I mean, it's easy, it's nice, inner cell mass and outer cell mass. I see what he's trying to do, but anyway, this is said that the trophoblast is said to give rise to extraembryonic tissue. namely the placenta which surrounds and kind of nurtures the baby along. Tropho actually means nurturing. All right, so here's what we have. Here's our little morula, and now we're looking a little deeper into it, and it is segregated. So we have an inner cell mass, and we have a trophoblast around the outside. Wonderful. Now where we get down a little bit, we're not going to go crazy into this, but we do have to know some of these names if you want to crush boards. If you don't want to crush boards, if you want to just get through boards, I don't think you need to know this stuff. But there's some of you who want to crush boards, and if this gets on YouTube, medical students have to know this, just period. Their boards are, for what I understand, tougher than our boards are. Anyway, so the inner cell mass, if you want to become an inner cell mass cell, you have to have two genes expressed. What does that mean, two genes expressed? That means turned on and running the central dogma. So that gene has to go through transcription and translation and make a protein. So what are the proteins that need to be turned on or made or expressed is the official word for that. Something called OCT4 and NANOG. OCT4 and NANOG. All right, so if you want to become an inner cell mass cell, these genes need to be turned on. And we'll see these do some stuff later. We're not done with those two. All right, these two gene products, once they're turned on, they also turn on two other genes. They can express factors which wake up other genes. And these two are really important. WENT, the WNT gene, I just call it WENT. And WENT is function in life is to turn on a very, very important gene and gene product called nodal. Nodal. We will talk a lot about nodal. All right. Now, what about to recap? To become an inner cell mass cell, you need to wake up OCT4 and NANOG. They're going to express some transcription factors and other things that wake up and turn on the WENT gene. The WENT gene, in turn, turns on the nodal gene. So basically, all these four need to be functioning, or you won't make it as a little fetus. Never make it to that stage. How about to become a trophoblast? You have to turn on two different genes, one called CDX2 and one called GATA3. Those genes need to be on. What do? They actually prevent OCT4 and NANOG from turning on, because if you turn on OCT4 and NANOG, you're going to become an inner cell mass, and we don't want that. We need to stop those genes from turning on. And so to stop them, we express CDX2 and GATA3. All right, and that makes, that inhibits it from becoming an inner cell mass. All right, so implantation is coming. We should talk a tiny bit about the parts of a uterus. You'll get this more in Gross 2, second quarter. But this is a uterus. It kind of looks like a pear. Here's the vagina. And remember, the job of the sperm is to get in the vagina and get up through the external os, the cervical canal, internal os, and get in here, get into the uterine cavity, and then make it up to the fallopian tubes to find that egg. But here are the parts of the uterus. We have a fundus, and then the fundus is just the top of the uterus, and that's it. The main part of the uterus is called the body. And we'll look specifically at this wall. Sometimes this is just called the uterine wall, but we'll look at the parts of that in a minute. And then we have the kind of gatekeeper, which is normally not open like this, but this is the cervix. These cells turn over quite rapidly, and when cells turn over rapidly, they're at risk for cancer, and that's one of the things females have to watch out for, cervical cancer down here. And there's some fornices. This picture isn't quite correct. There's these little cavities right here called there's anterior, posterior, and lateral fornices. You can look at that next quarter. I'm not worried about that. So the wall has three layers going from outside to inside. We're going this way. Let's talk about the outside. That's the perimetrium, and that's like the serous layer as well as an outer layer. You could call it that. It's made of visceral peritoneum or visceral peritoneum. Tomatoes. And then, so that's not very exciting. The big thick muscular layer is called the myometrium. This is important because it's highly vascularized. There's lots of blood vessels, and the little growing morula needs to tap into those. It won't be called a morula for long. We'll just call it the embryo. The embryo needs to tap into those in order to get blood so it can grow and survive. So this is where its goal is. Sorry about that. My frog is back. Let me get some water here. Okay, so it has blood vessels, lymph vessels. Yep, and it grows like crazy during pregnancy. And then we have the inner layer. This is the endometrium. So here's kind of a, well, it doesn't show the outer layer, but myometrium. Here's the martini glass would be in here, the lumen. There's our little morula. And the job of the morula is to implant right here in the endometrium. Specifically, the endometrium is broken down into 2 layers. So we're still going outside to inside. So the next layer would be the basal layer, or stratum basale, which is not the greatest word, right? Because if you studied skin yet, you know that that's the kind of the bottom germ cell, or not germ cell, but there's stem cells of the skin. The bottom layer is called stratum basale. And then the next layer is called the functional layer, or the stratum functionale. Right, the basal layer, it's adjacent to the myometrium. It contains basal cells called stem cells, the same kind that are in the skin that also has basal cells. And they give rise to cells that make up the stratum functionale. Because the stratum functionale, this layer right here, females lose this every month. This is what sloughs off at the end of your cycle, and that's your period, is made up of all this stuff right here. The stratum basale layer, that does not go with it. And why do you bleed when you have a period? Well, you have lots of blood vessels in here. So as this sloughs off, you break these blood vessels loose and you bleed. All right, so basal layer is not shed during menstruation. It contains the uterine glands, which are really important. It also has a stroma that is kind of the substance of the layer. So probably won't ask you much questions. This is Dr. Doe's area here. Functional layer, stratum functionale. We said that shed during menses. That's a girl's period, is called menstruus or menses. And it does have an epithelium on it, and it's made of simple columnar, according to Junqueira. That's your histology book that Dr. Doe hopefully is using. That's the board book for histology. And yep, so these are weird. So we have a whole bunch of these. You know what these cells are, right? They're columnar looking, like that. All right, here's the martini glass. So this is the endometrial fluid here. There's our oocyte coming. But there's 2 types of these simple columnar cells. Some of them have these little cilia, these little hairs growing off them. And some of them just have a little hole here, and they secrete juices up here. So those are the secretory cells. So we're going to come back to those, because normally these secrete a really slippery fluid that eggs that are non-fertilized will hit, and they can't stick. We don't want unfertilized eggs sticking and binding to these endothelial cells. So if that's not fertilized, it gets on this little slippery stuff, and it gets kind of swept right away. And the cilia kind of push it away and keep it from sticking, because we don't want any implantation. Only these little fertilized oocytes get to stick here. We'll see how that works here in a second. All right, there's just a review, another kind of a coronal section. There's the cervix. Now we can see the fornix, lateral fornices here. Cervical canal is here. There's the external, internal os, uterine cavity. We talked about there's a little sperm trying to get up there to find the egg. All right, so let's go back to our story. So we had a morula. Let's That was day 4. Now day 5, by day 5, that little 16-cell morula becomes a 36-cell morula. Hang on one second. There we go. My headphones were dying. All right. So by day 5, our little 36-cell, or by day 5, we've divided so many times that we have a 36-cell morula. And so our trophoblast is bigger. Those new cells, some of them became trophoblasts, some of them became inner cell mast cells. They're still segregating away. But on day 5, it comes out of the fallopian tube and drops into the martini glass, which is the uterine cavity. So the job of the morula is to go over and stick to that epithelial layer of the endometrium. All right. So it also turns out that now we're floating around in fluid. This thing is not watertight, so it's not the greatest ship in the world. Let me see if I missed anything. Let's see. Yeah, the hormone progesterone. Remember that corpus luteum that we talked about, I think, on week 2? That used to be the follicle and was converted into corpus luteum. It's secreting things like progesterone. And part of its job now, the progesterone that it secretes relaxes the fallopian tube sphincter here, so this morula can pop out. If something goes wrong with the corpus luteum prematurely and progesterone isn't secreted, the little fertilized egg can't get out of the fallopian tube and will actually grow in the wall of the fallopian tube. It's called an ectopic pregnancy. It's complicated, isn't it, all this stuff? So there's our morula. It's a leaky ship. It's sinking. Why is it taking on water? You'd think it'd be airtight because it has a zona pellucida around it, so it shouldn't be leaking. Well, it turns out we have another little molecular biology story that turns on. So the genes that make sodium potassium ATPase pumps, which you've probably talked about or you will talk about in physiology, they're born. And so our little zygote has sodium potassium ATPases pumps going by day 5. And when it drops in the martini fluid, or the uterine fluid, it starts sucking in sodium. And if you don't know, wherever sodium goes, chloride usually follows, but water always follows like a puppy dog, except there's some exceptions to that we'll talk about in 5th quarter, and you'll probably talk about with renal physiology. But yeah, in exchange for sodium coming in, potassium gets kicked out. And so who does that? The sodium ATPase. So bottom line is we're filling up our little morula with water, and that starts to carve out a little cavity right here, and it pushes the inner cell mass up to one side, and then it pushes the trophoblast cells. I mean, they still stay to the outside. So we have a cavity. How exciting. So there's, much better than I drew it, there's a cavity, and that has a name, and that's called the blastocyst cavity. You definitely should have, oh, there's a star. I was going to say, you should put a star. I always ask that question. Those of you who, what do you study? There's so much material. Well, there's one. Just to make a note card. That's going to be, you have to know what the blastocyst cavity is, aka is blastocoel. And that's going to actually morph into something called the primary yolk sac. Primary yolk sac is going to morph into a secondary yolk sac. Okay, eventually this will be crushed by another cavity called the amniotic cavity, which we'll look at here in a minute. But so far, this is our morula. And we're not going to even call it a morula anymore because a morula doesn't have any cavity inside of it. So we have to name it something else. So because there's this blastocyst cavity or blastocoel, why don't we just call it the blastocyst? And that's exactly what our morula becomes. So day 5, say goodbye to the morula. It becomes a blastocyst. When in doubt, call it a zygote. Although that won't help you on the test probably. Let's see. So here's a blastocyst, a human blastocyst, and blastocyst cavity. You can see it nicely, aka blastocoel. You can see the inner cell mass has gotten quite big, and you can see that we have the trophoblast around, surrounding all this stuff. Okay, and we still have a zona pellucida. You can faintly see it here. We still have a plasma membrane. We're going to get rid of the zona pellucida next. Oh, one more concept here. So the embryonic pole, if this is the world, this would be the North Pole, this would be the South Pole. But it's not the world. But wherever the inner cell mass kind of conjugates, that's going to turn into the embryonic pole. So this is the embryonic. I'm going to write that out because my fingers don't write very good. Embryonic pole. All right. This is the AB, abembryonic pole. Let me just go to the picture. All right. So there's the embryonic pole, abembryonic pole. What's the embryonic pole? That's where the cell mass, or the inner cell mass, lives. Okay. Where's the abembryonic pole? It's exactly opposite to the embryonic pole. It's 180 degrees away from the embryonic pole is the abembryonic pole. Right? Pretty simple stuff. There's another human embryo. It's developing. You can see a huge blastocyst cavity. You can see the trophoblast. The zona pellucida is getting thinner. So the cell membrane is still hanging in there. The other layer is gone. The corona radiata is gone. We lose that about day 2. Hatching. Okay, did you know that we hatch? Every one of you still listening, you have hatched at one time. And so hatching occurs when our little embryo leaves the zona pellucida. And that zona pellucida, I mean, that thing is still pretty tough looking. So we need to get out of that. You can't implant into the uterine wall with a zona pellucida. You need to get rid of that. And so late in day 5, the blastocyst hatches from the zona pellucida. Now we're not exactly sure what happens here because we have such a limited number of human embryos to study, so they don't know for sure. But it's believed that some protein or some proteases are released, probably from the inner cell mass, and it dissolves because hatching occurs at the embryonic pole. Above the inner cell mass is where it occurs. So we don't know the full story. But after a hole is made, the blastocyst is kind of ejected and gets out, and we don't even understand the whole mechanism behind this. How does it get out? Is there smooth muscle? We don't know the story. But we sure have nice pictures of it. So you can see the zona pellucida is left behind as the blastocyst takes off. And so that's hatching occurs at the end of day 5. And now we have a naked blastocyst. Some say the plasma membrane is there, still hanging in there, and some say it's gone. So we're even not 100% sure. But by the time implantation comes, that will be gone. So very shortly, that kind of dissolves away. All right, so basically, question? It's thinner. It's gotten thinner. It's kind of degenerated away, but it's still, that's a good question. It's still, I mean, there it is. We're looking right at it. It's still pretty beefy looking. So that's why they think it's got to be some type of protease that cuts through it, because I don't think you could poke through that. But it's that absolutely. That's a great point. It sure does thicken up, so polyspermy doesn't happen. So that's why we think it's got to be enzymes that eat a hole through it, because I don't think you could get through if it was nice and strong like this. All right, so our naked blastocyst, is our new word, is loose. What happened to the corona radiata? I said a couple times. Day 2, that corona radiata just kind of degenerates away. There's no hatching involved. This degenerates away. But because of that stiffening, that zona pellucida is pretty strong. So That one hangs around until hatching occurs. Alright, so now we need to talk about some more molecular biology. Some more segregation occurs. And this time it's the inner cell mass that is going to divide. So right now we just have one big ball. Where is it? Where's a picture? There's one. So right now we just have one ball here of tissue. Let's segregate that as well. And that's what happens next. So we are going to create the most important group of cells ever. So the epiblast cells. So the inner cell mass, let me draw it here. So here's our inner cell mass. It's going to split into two groups of cells. It's going to split into these epiblast cells and hypoblast cells. I'll make those three. Okay, hypoblast cells. So we're basically splitting these apart. These are the ultimate stem cell. These can be converted if you could harvest these things. They turn into anything you can imagine. Your eyeball, your heart, your toenail, you name it. You could make it with this cell. Super important cell. And then we're going to get some hypoblast cells, which are important as well too. And we'll keep talking. Now we're going to convert these into an ectoderm and an endoderm. So these are going to be converted into a more functioning type of cell. But nevertheless, this is where ectoderm and endoderm come from. These epiblasts and hypoblasts. And we're not even implanted yet. So this thing is getting the ability to turn into a human already, and it's not even implanted. Histologically speaking, these cells also take on a more columnar look, and the hypoblast cells take on a more cuboidal look. Alright, so there's what we have now. So there's our blastocyst, and we have epiblast cells, and we have hypoblast cells. So we have different layers have been created already. We have kind of a we talk a lot about the trilaminar disc. This would be a bilaminar disc, which is what we're making so far. So now the question is, so that's easy, but the question is, how does that happen? So we need to talk about FGF4, or fibroblast growth factor 4. It's another gene that needs to be woken up. And in epiblast cells, the gene for FGF4 wakes up. It gets turned on. We don't know how it wakes up, but it wakes up. And that's the gene that tells them to kind of move up to the top, closer to the embryonic pole. All right? And at that point, once they move up there, they become epiblast cells. So, I guess we do know what wakes it up. So it turns out that FGF4 was woken up by NANOG. Okay, so we talked about NANOG already. That was the first gene turned on. So NANOG actually turns on this super important FGF4. So FGF4 expression, so that's the central dogma. Expression just means going through that transcription, translation, and making the FGF4 protein. That's what that means. And that was induced by NANOG. All right, so how does that work? Let's see, how does it do that? FGF4 from epiblast cells also diffuses into the adjacent cells and stimulates them to express GATA6. So the hypoblast cells, that's what I should have said there. How's the hypoblast story? We know the epiblast story. We know that NANOG woke up fibroblast growth factor 4, and that caused them to clump up toward the embryonic pole. Well, what about the hypoblast cells? What's the story there? Well, the FGF4, once it's on, it actually soaks into the cells below and causes them to express GATA6, but not them. They don't express GATA6, but the cells adjacent to them, because of FGF exposure, express GATA6. And with GATA6 on, they're instructed to stay below the epiblast cells. Okay, so that kind of splits them into layers. So in other words, FGF4 expression equals epiblast cells and GATA6 expression equals hypoblast cells. I think I got some pictures of that. So here's kind of the situation and why some just have FGF4 turned on and others kind of become a slave to FGF4. Don't understand that. But in this cartoon, all the blue ones have FGF4 on. So, and they must have turned on first, and it soaked into the ones next to them and turned on GATA6. Once GATA6 is on, these guys are destined to become hypoblast cells, and the blue ones are going to be epiblast cells. So in a few hours later, after this picture was taken, you get a nice differentiation. So there's epiblast cells, and there's hypoblast cells. Hypoblast cells have the GATA6 gene on. Epiblast cells have the FGF4 gene turned on. Okay, we'll just pretty simple, straightforward. And yep, so we have kind of a bilaminar disc starting. That's you. That little thing there, that's going to be you right there. Not this stuff. This is going to become your placenta and help you develop. You're going to lose that at birth, but this is you. All right, so we basically have met the bilaminar disc. As we said, it's a two-layered, the inner cell mass turns into a two-layered bilaminar embryonic disc. Sometimes it's called the bilaminar disc. Sometimes it's the bilaminar blastoderm, which I don't like that word, but that's from European books. But yeah, there it is. And now, once we've established this setup, which you can see under electron microscope, we also have established the dorsal ventral axis of the body. So we know this stuff is dorsal, and we know this stuff is ventral, right? Because the north pole, the embryonic pole, is up here. So if you're close to that, and the south pole or the abembryonic pole is down there. So we basically know up from down. We know dorsal is here, and we know ventral is here. So already that said that the dorsal ventral axis has now been established. All right, everything I've already talked about, ventral and dorsal, awesome. Okay, let's do some more differentiation now. This is a very important area, these AVE cells, the anterior visceral endoderm. So another segregation story happens. So the hypoblast cells in the cranial region, so we don't know what we know dorsal and ventral, but we don't know the cranial from caudal yet, but we're going to in a minute because of this story right here. So the hypoblast cells, remember these are hypoblasts, that's the bottom layer. So some of these cells get some genes turned on, and they're going to become another type of cell. So the hypoblast cells in the cranial region, some genes wake up, and they're going to become what's called the anterior visceral endoderm. And it's the same kind of story. So to become an anterior visceral endoderm cell, we have to turn off nodal. Remember, nodal was turned on in our first step. That's a really important gene. So we need to turn that off in the hypoblast cells to make AVE cells. And it sounds easy, but it's not so easy to do. This is a very powerful gene, and it doesn't like to be turned off, so we have to have quite a bit of help to do this. Recall nodal was turned on when the inner cell mass first developed. Yep, by turning on OCT4 nanog, that first story I told you, turned on nodal. So how do we get this thing turned off? Well, the most important gene, the nodal turner offer, of course we don't call it nodal turner offer, it's the nodal antagonist. And it's a terrible name, I guess, Dickoff-1, named after the scientist Dickoff, who that's like the worst name. I think I would change my name if it was Dr. Dickoff. But nevertheless, that's his name, and it's abbreviated DDK1. And he was the one who discovered this gene. And it gets turned on in the cells that are going to become AVE cells. If you don't turn on DKK1, you can't become a anterior visceral endoderm cell. So Dickoff-1 needs to be turned on. We're not sure what turns it on, but once it gets on, it's able to block Wnt. And we know that Wnt is kind of the henchman for nodal. You need Wnt functioning to turn on this powerful nodal gene. So we can't flat out turn off the nodal gene, but we can turn off the guy who turns on nodal. All right? So Dickoff-1's turned on and it blocks the expression of Wnt. And that helps, that does it, but it turns out it doesn't, it needs help because nodal is so powerful. So we need some other guys to help turn off nodal. So there's other genes that become turned on in these future AVE cells. Lefty1, sounds like gangsters. Cerberus1 and Lefty1. They also get turned on, and so they help turn off nodal and Wnt, and we won't go any deeper than that. So the recap is DKK1, Dickoff1, Lefty1, and Cerberus1. They all have to be functioning if you want to be an AVE cell. So with all those three genes turned on, the nodal gene and Wnt genes are both turned off. Okay, great. So what? So here's the story. So here's where we were, right? We had the epiblast and the hypoblast in yellow, but these genes all of a sudden start expressing Lefty1 and Dickoff1 and Cerberus1, and that made them special. So they turned into these little red ones, and these are AVE cells now. And now once that happens, we can see this under electron microscope. Once this is established, now we know this is going to be the kind of the head end or the cranial end of the little creature, and this is going to be the caudal end of the little zygote or blastomere or embryo. Okay, so we've established the dorsal to ventral axis, and now with the development of AVE cells, we've established the cranial caudal axis. And that's a nice, fairly easy board question. What group of cells come into being, or what group of, the expression of what group of cells establish the cranial to caudal axis of a blastocyst? And it would be the AVE cells, because once they pop up, we go, oh, okay, that's going to be the cranial end, right? So we made a big deal about these AVE cells. They better do something, and they do quite a bit of stuff. We'll talk about next time, a primitive streak is going to start to form up here, or it's not going to form here. A primitive streak is going to start to form back here. And we don't want the primitive streak forming up here, or you'll have a little monster, right? You will have a non-viable fetus. So what does these AVE cells do? A bunch of stuff, but one thing, they inhibit the formation of the primitive streak above them. You get all sorts of mutations if that occurs, and usually the fetus is non-viable. How do they do that? How do they stop the primitive streak from forming? Well, again, the AVE cells, we know they secrete the three nodal inhibitors, Cerberus, Lefty1, and DKK1. Those are the nodal turner-offers. Turns out these also soak into the cells above. Remember, we have epiblast cells above, which I kind of messed this picture up. But these would soak up into here. The nodal turner-offers would soak up here. And once they soak up there, it inhibits those cells from, it does the same thing. It inhibits them from expressing nodal as well. So if you don't express nodal, you can't form the primitive streak if you're an epiblast cell. If you're a hypoblast cell and don't express nodal, you're going to turn into an AVE cell. Okay, I know this is complicated. Go back and listen to it. This takes a little, make some note cards to get this story down if you want to crush things. But this is, you got to know this stuff, especially if you want to crush boards. All right, so they diffuse into the cranial epiblast cells and inhibit them from making nodal. And without nodal, you can't make a primitive streak cell. Right? What else do they do? They're also really, if that's not enough, they do more. So these AVE cells, they turn into these super signaling centers with all kinds of different genes on, and they are able to tell cells what to do that pass by them. And they basically, the early formation of the heart is all about these AVE cells. So they build the very complicated heart. The caudal AVE cells, in addition to helping build the heart, they do something else. They build the precordial plate, which we will talk about. Precordial plate becomes a super signaling center, which is very important in formation of the head and perhaps more importantly the forebrain. So basically AVE cells are indirectly responsible for forming part of the brain and the heart, and they make sure that the primitive streak doesn't turn on. So very important cells. All right, let's get our, we haven't even bound yet. I don't think we're going to make it through all these slides today, but let's go a little further anyway. Uterine preparation. So we talked about binding and that the slippery stuff that the endothelial cells of the endometrium secrete. Turns out that slippery stuff has a name. It's called mucin 1. So the presence of a developing blastocyst that's gotten into the uterine cavity, it stimulates those epithelial cells, the secretory epithelial cells. Remember, there's cilia. Some of the epithelial cells have cilia on the endothelial lining of the endometrium, and some were secretory. So it turns out, and we don't understand the mechanism completely, but it shuts off that slippery stuff production. And that, so mucin 1 is the slippery stuff. If mucin 1 is flowing, even a fertilized egg can't implant. You need to shut off mucin 1. And so just the presence of the blastocyst in the martini glass shuts that down, and we don't have a good story on how that happens. It is a glycoprotein, and it makes the endometrial lining slippery, as we said. And with that gene off, now a fertilized oocyte, we'll call the blastocyst, is where we're at now. It can bind to the endometrium. All right, and the binding happens usually about day 6 or day 7. And with the mucin 1 off, the embryonic pole again of the blastocyst is the one that binds to epithelial cells of the endometrium. Those epithelial cells, we got to know who the receptors are. So the binding is accomplished by receptors on the trophoblast of the blastocyst. Our little fertilized ball has off the north pole, it has some receptors called L-selectins, and they hook up with receptors called trophonins that live on endometrial cells, and those guys hook up. Let's look at a picture. So there is our blastocyst. This is our embryonic pole. We know. How do we know that? Because the inner cell mass lives at the embryonic pole. We have an epiblast and hypoblast layer. I could have drew the AVE cells. I guess the AVE cells are maybe right here. But nevertheless, there's the L-selectin receptor, and it binds to this trophonin receptor that is on the endothelial cell. And it's as simple as that. Mutations in the genes that make L-selectins or trophonins, you're not going to be able to get pregnant. So that's another really important, and there is only one set of these that is currently known or accepted. So this is, remember the sperm and the zona pellucida, there's a couple different receptors for ZP3, but there's only one that we know of so far for this. Let's do a little rabbit hole, ectopic pregnancy. Because normally this implantation occurs on the posterior wall of the uterine cavity. It's okay if it's on the lateral walls, maybe even the anterior walls, but there's some places you don't want this implantation, or you don't want this hookup to occur. And that's called an ectopic pregnancy, when the hookup occurs in places where it's not supposed to occur, like in the fallopian tube. There's no way you can grow a gigantic baby in the fallopian tube. It's just not going to happen. You'll rupture the fallopian tube, horrible, horrible pain, and you'll bleed, and it can be quite dangerous. Strange parts of the uterus, it can't happen. The ovary, the peritoneal cavity, for goodness sakes. Let's look at, and these are all called ectopic pregnancies, and they are life-threatening to the baby and mom. And I will ask this question, so make sure you know at least these 3 most common places for ectopic pregnancy to occur. Number 1 place, surprise, 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 the same place fertilization occurs is the ampulla of the fallopian tube. That's the #1 place for a misimplantation to occur in ectopic pregnancy. Now, there's no way you can grow a baby. You will explode that fallopian tube, and that will cause horrible, horrible pain and bleed like crazy. And we'll talk about a Cullen's sign when we get in 5th quarter. You can see blood around your, kind of like a bruise, you know how the skin gets purple? You can see that circling around your belly button, and that's often the sign of an ectopic pregnancy that's ruptured something. #2 is the isthmus of the fallopian tube. That's the second most common implantation. Should not occur there. And #3, it's up here at the fimbriae, which still shouldn't occur because you can't grow a baby there either. There's some rare ones. You could get one on the ovary if the oocyte comes out and the fimbriae miss it. The ovulated oocyte may stick to the ovary, and the sperm will find it. Remember, they're like hound dogs. They'll hunt that thing down, and you can get a pregnancy there. You can get one. Let's see, did I not have it on here? Oh, there it is. So it's super rare, 0.2% of the time, but you can get one in the abdominal cavity or in the pelvic cavity, right? There's the pouch of Douglas there, or the retrouterine pouch. You can get one there about 1% of the time. So they can be inside the peritoneal cavity, or really in the pelvic cavity if you want to get specific, right? And then cervical implantations rare, 2% of the time up here at the top at the fundus area. I don't know why they call that. Should be fundal, really. I don't know why they're calling that interstitial. It should be the fundal area. All right, but know these top 3. I mean, you should know them all, but for sure know those top 3. Okay, let's meet a new cell type, and then I think we'll call it a day. We have something called the syncytial trophoblast. I always talk to my 5th quarter students. I say, what do you remember about embryology? And they go, oh my God, one word we know, syncytial trophoblast. They seem to memorize this word. It's kind of a cool, name your dog syncytial trophoblast. So what's the story with this? So after implantation occurs, our trophoblast, our outer layer of cells, they start to proliferate by mitosis. They go through mitosis and they make a new crazy type of cell. And this is a weird cell that has no cell membrane. They share a cell membrane, maybe 10, 20 cells. They all have nuclei, but they lose their cell membranes and they share a group cell membrane. Really weird, right? Almost like a cancerous type thing. That's called the syncytial trophoblast. Let's take a look at it. Right? So here's the trophoblast see, this is a mistake. They shouldn't call it that yet, but we'll get to that. But there's the trophoblast layer in green, and it's growing new cells. The cells are thickening here, but then it's growing these weird cells. Here's 4 cells without a cell membrane. They share a common cell membrane. So that's pretty crazy. And that, in light green, that's the syncytial trophoblast that's starting to form. All right, so although they have a nuclei, as I just said, they share a cell membrane. They're highly invasive. They grow like wildfire, almost like a cancer. And the leading tips will push their way through the epithelial cells and into the stroma and start kind of like a cancer invading the endometrium. Right? How do they invade? They have some weapons. They secrete these proteases called matrix metalloproteases. We'll actually expand on this in spinal anatomy. We'll talk about the disc, or how the nucleus, how these things are important, and how imbalances can cause trouble. But those enzymes will cut right through the endothelial cells. All right, so they're powerful proteolytic enzymes, and they literally drag our little fertilized egg. They drag it inside the endometrium. All right, so this picture, they're invading. They're secreting MMPs, and some of those stromal cells are kind of put into an apoptotic state where they die. So not only do they cut, but they kind of paralyze the cells they're going through. And yep, as they grow and grow and grow, they pull the blastocyst deeper and deeper and deeper. And the function of these cells is to find mom's blood supply, because they're going to need to tap into that, because only so much cell growth can occur without blood. And here's mom's blood vessels. So the syncytial trophoblast has grown much greater. It's actually there's mom's blood vessels here, so it's starting to get into mom's blood vessels. And then we're going to need to talk about how a circulatory system is set up in this, because these are cells. They need blood to stay alive. What else do these syncytial trophoblast cells do? Well, here's your pregnancy test. They also secrete human chorionic gonadotropin, which is HCG, and that is a common pregnancy test to tell if you're pregnant or not, because normally females don't have HCG flowing through their bodies. There's another pregnancy test that even more earlier, but it's not. This one's quite accurate. So HCG is very important. What does it do? Besides pregnancy test, I mean, that's not the job of it, really. Remember, our corpus luteum normally would die, and so the HCG extends the life of the corpus luteum to keep it alive. Corpus luteum is secreting progesterone and estrogen, and it needs to do that for about 12 weeks. By 12 weeks, the placenta will be formed, and it will take over the job duties of secreting progesterone. Estrogen's not quite as important at that point, but we need progesterone. And so HCG keeps the corpus luteum alive for about 12 weeks, so the placenta can develop and take over that job. All right. And there's, if you forgot what the corpus luteum, remember we went through that story in week 2, and there's ovulation occurred, and there is the corpus luteum, which is secreting progesterone and estrogen. All right, that's probably, yeah, let's stop right there. That's probably enough for your brains. All right, I'm going to stop it at slide 61. Let me shut this down.